Okay, so the first safeguard against errors is the fact that the active site closes around the substrates, and so the catalytic reaction can only occur usually when we have a Watson-Crick base pair bound in the active site, because that's the only type of base pair whose shape is accommodated by the closed active site. The second safeguard is editing. We edit our mistakes. And then the third safeguard is something called mismatch repair that gets us the rest of the way there towards that 10 to the minus 9th error rate. Okay. And so here's a mismatch. Okay. And so the question is, how does, so let's say a mistake arises. Despite those safeguards about, one time in a million, a mistake is still going to, DNA polymerase is still going to leave behind a mistake. Okay. The question is, how does the cell recognize which strand contains the mistake and therefore which strand needs to be repaired? Well, we don't really completely understand how this works in eukaryotic cells, although there's some ideas. In bacteria, though, we understand that very well. The way that the bacterial repair machinery knows that there's which strand contains the error, and therefore which strand to repair, is by looking at the methylation state of the strands. So in bacteria, in E. coli, for example, we have a kind of methylation called DAM methylation. It just stands for, I'm not saying curse word, it stands for deoxyadenosine methylase, right? So, or deoxyadenosine methylase, okay? And so what we have on these adenines then is an extra methyl group attached to the N6 amino group, right? So, and so this enzyme then adds these methyl groups to these adenines, but it only adds the methyl group to the adenines when they come in this sequence context, GATC. Okay, and this sequence occurs randomly, roughly every 250 base pairs in, a typical bacterial genome. So it occurs quite frequently. Okay, and the important aspect of this sequence is that it's a self-complementary sequence. So wherever we have GATC on one strand, we also have GATC on the other strand. Okay? And so before DNA replication occurs, this site is in its fully methylated state because DAM methylases come along and put methyl groups on both of these adenines. But then after DNA replication occurs, the DNA is only hemimethylated. Because the DAM methylase hasn't had a chance to get in there and do its job, right? And so the newly synthesized strand will be unmethylated. The parental strand, the mother strand, will be methylated. Okay, and so what the repair machinery looks at to determine which strand is the newly synthesized one, and therefore which one is likely to be the one with the mistake when it sees a mismatch, is which one is unmethylated. Okay. Okay, so this figure from your book shows how that works. Let's say we have a mismatch in the DNA. Just after DNA replication, then, the parental DNA is methylated. The daughter DNA is still not methylated because DAM methylase hasn't had a chance to get in there and do its job. What happens is the two enzymes called mut-S and mut-L recognize the mismatch and bind to the mismatch. So they just bind to any old mismatch. Okay, and then they begin translocating the DNA through this mut-L, mut-S complex, so we get a loop here that contains the mismatch. Okay, as that loop forms, then you reel in the flanking DNA, and eventually the mut-L and mut-S come into contact with a GATC sequence, a hemimethylated GATC sequence. So we're calling this hemimethylated because it's half methylated, right? Okay. Once it comes into contact with this hemimethylated GATC sequence, that triggers the recruitment of another enzyme called mut-H. Mut-H is actually an endonuclease, and what it does is it looks at the GATC sequence and it specifically cleaves the unmethylated strand. Okay. Now, a type of enzyme called a helicase, we'll talk a little bit more about what helicases are next time. An enzyme called a helicase comes in and unravels this DNA from the nick here all the way to the mismatch, and then a nuclease, an exonuclease, comes in and degrades the DNA all the way from the nick to the mismatch. Okay? And now DNA polymerase can bind to this end and basically try again and hopefully get it right this time by filling in the gap with the correct nucleotides. Okay, so this system catches 99.9% of the remaining mistakes so that the overall error rate of DNA replication is 10 to the minus 9th. So very similar enzymes are involved in the mismatch repair process in eukaryotic cells, in humans, for example, and it actually turns out that mutations in the mut-L and mut-S homologs in human cells can result in a genetic predisposition to cancer.